Basically, the topic is clinical research, concept, conduct, and significance. In this, basically, this is going to be a very introductory, very superficial kind of a lecture, not very deep, not going to talk of very big things. Going to talk very superficially and going to introduce, basically, we'll be introducing the terms that you may encounter during the research. And the talk is divided into three parts. The first is the concept, as it says, where we'll be talking about what are the various designs and how they are classified. Conduct, that how a clinical trial or clinical research is conducted. What are the regulations governing the clinical trial and conduct? And lastly, we'll be talking about significance, where I'll talk about other things which are related with the conduct of clinical trials. The first slide is discovery by forethought and serendipity. I don't know how many of you have read the Amir Khusro's Hasbishit, which was written in 1302 and later on translated, where this is basically later on in English translation is known as the Three Princes of Serendip. This is a story of three people who have power of deduction, and by looking at things that are happening, they are able to decide what it has. So basically, their father wants to choose the best among the three. He sends the three princes to a far east land. They are walking in the desert, and then they see a camel footprint in the desert. The one says it's a camel. The second says the camel has one eye. Then the first again says the camel is carrying honey and oil, and then like this. So it's about deduction, and discovery by forethought and serendipity basically is the faculty of making discovery by accident or unexpected observations which you encounter during ongoing investigation leads to another discovery. So basically, it deals with the power of deduction. We talk a lot about luck, and scientifically, the luck is classified into four different type of luck. The most common or most rare is the blind luck. Blind luck is something which provide opportunity to anyone who is motivated to research, and just like that, a rare disease, rare clinical presentation, or rare phenomena occurs in front of your eyes, and you are there to witness it. Things that happen by chance are type 1. Type 2 is motion. Motion is a type of luck which happens when you are looking for something else and you stumble onto something new. The example which is given here is of the Epstein-Barr virus. Burkitt was working with malaria, and he encountered what is today known as Burkitt's disease. So what he did was he thought that it is also a vector-borne disease, just like a malaria. And when he came to England, he met Epstein, and who did that was the time when scanning microscopes has just come, and he found out that it is a virus particle which is causing it. So because he was working on malaria, he observed a disease, and a chance meeting with Epstein led to discovery of Epstein-Barr virus. This type of deductions are called, or chance are called, motion. He was not looking for Epstein-Barr virus. He was not looking for the Burkitt's disease also, but he was working on malaria. So this disease was detected while he was working on malaria. So it's called motion. The third is called recognizing good fortune. The example here is of Alexander Fleming and Florian Chain. Fleming discovered the penicillin, but it was Florian Chain who found out that what was the importance of penicillin, that it kills bacteria. Opportunity which is looked by all but one. So it is something which Florian Chain found, and it was a class 3 chance that what even Alexander Fleming could not discover, Florian Chain did. And the fourth, which most of us do, is persistent and lateral thinking. We keep doing more and more research, and we keep thinking, Every time we read a paper, every time we go through something, we keep persisting and go for a lateral thinking out of the box. What is called is the directed motion. Research is, it's a very simple definition of research, that research is an organized and systematic way of finding answers to question. That it is systematic, that is, it uses a definite set of procedure and steps that are to be followed. It is organized, that there is a structured methods in going about these things, and the questions which are essential to central of the research. I'm not going to talk about how to frame questions, how to convert your questions to hypothesis, how to convert your hypothesis to null hypothesis in this particular lectures. But if later, if somebody thinks that they should learn that, probably another lecture on that we can organize. Clinical research is a branch of healthcare which determines the safety and effectiveness of medication, devices, diagnostic products, and treatment regime which are intended for human use. So when the research is applied to the clinical science, it becomes a clinical research. This can be for prevention of a disease, can be treatment of a disease, or diagnosis of a disease, or relieve symptoms of a disease. Whatever it is, when it is has a clinical application, it becomes a clinical research. There are different classifications given for research. I'm going to use only two. And the simplest classification is observational and interventional. Observational clinical trials are where you simply observe the cohort or the people, group of people with the similar characteristics, and do nothing. Just keep recording your observations. Those studies are called observational studies. And where you intervene by meaning of doing a test in one and not doing a test in the other group, or giving a drug to one group, not giving a drug to other group, or giving a different drug to other group, it becomes an interventional trial. And the interventional trials can be randomized and can be blinded. Simple classification: observational and interventional. Look at the kind of studies that we do. Most of the descriptive studies that we come across are observational studies. There are some which are called cross-sectional studies, and the case studies and the case reports that we publish are all type of descriptive studies. There is no intervention being done in most of them, but case reports or case studies can be interventional also. And interventional studies can be randomized, blinded, double blinded. There can be single group studies where you use only one group of patients and single drug, or different doses of drug in a single group. You can have placebo controls, or you can have crossover trials. Crossovers, when there are two treatment arms and patients are allowed to cross over from one treatment arm to another treatment arm, is called crossover studies. And in the bottom, there is something called as a social science research, which I will not be talking about in this particular presentation because it's an entirely different form of research, uses different designs, different tools for measurements. And this is basically used for something like quality of life studies, like if you want to measure cost masses, you want to measure burnouts, then what the research you do is a social science research. Even the cost based research, if you want to see cost effectiveness, those are also comes under social science research. Brahman and as Bell has divided it into exploratory research, what we also called as pilot studies. This is a research which is done before there is enough evidence available. So we have a question, but we don't have enough material to go about. We don't know how to carry out a sample size or how to calculate a sample size. You can take example of say corona spread. There is no background information on how infective the virus is, what percentage of people has to be infected before a herd immunity develops. So what you do is you do a exploratory research first, do a pilot study, see what is happening, and based on the results of the pilot, you design your final studies. The advantage of doing exploratory research is it helps you determine the best research design, data collection and method, and use of subjects. If you don't do a pilot and straight away launch a study, you may be successful or you may not be successful. depending upon whether the design you took was the right design or the wrong design. Then it has descriptive studies. Descriptive is basically which describe the characteristic of a population. So basically, there are 5 questions: how, when, why, what, and who. These are the 5 questions that a research has to answer. So descriptive addresses what and who. That means it will tell you that what kind of subjects will benefit or what kind of subjects will not benefit, and who will benefit, who will not benefit. But other questions it's not going to answer. So this is a descriptive study. It describes the characteristic of a population or a phenomena being studied. As you are reading every day about COVID, so let's take an example from COVID. So when we say that the COVID, what are the group where the mortality rate is high? So you take all the people who have died from COVID, look at the age groups in which the deaths have occurred, and most of the time you're going to find that it is the elderly people who are dying. So this is nothing but a descriptive study where you looked at all the deaths that has happened in the world because of COVID, and you simply stratified those deaths by age. And at the end of the day, you find that uh, which population is or who is dying from COVID. So it does not establish a causal relationship between variables because you cannot study any more variables. You can simply say that what is happening and to whom it is happening. On the other hand, if you have why and how questions, then you have to look at the do the analytical studies. In an analytical study, you basically look at the cause-effect relationship among variables. You can have a single variable, you can have multiple variables, and uh, you can use analytical methods like statistical methods, and you can model your data based on uh, these procedures, these tools. So this is called the second tool. It's called analytical tool, which is going to tell you that why and how the things are happening. Another research strategy which has come into force, and I'll talk about it later. It's called critical. The critical basically has come from Karl Marx critique. It basically is a research which challenges the norms or the evidence which is already established. And this kind of research is sometimes called as transformative research because it is going to transform the already established theories and by challenging them. I'll talk about it, that why it is done and how it is done. Basically, it focuses on analysis of bias, inconsistency, gaps in knowledge, contradiction in account, theories, studies, or model. So if you have established theory, it basically challenges, tries to find out that why this theory is wrong or could be wrong, and then challenges and designs a new study which challenges the established studies. Then most commonly that we do is predictive and confirmatory studies. This is the most commonly done studies, which are quantitative studies, which identify measurable variables, and we can manipulate the variables to produce measurable effects. Here we use a null hypothesis, which is proven or disproven, and it is dependent on accurate sampling and uses the uses statistical tests to establish the causal relationship variance between sample and the predictive trends. So this confirmatory and predictive research is something which is commonly done. Then there was a question last week on number needed to treat. Uh, so this is for Tarun especially. Number needed to treat is the number of patients you require to treat or prevent one additional bad outcome. The flowchart is given here. Suppose we have the example. I'll read the example rather than the what should be done. The example used here is a clinical trial of cholesterol lowering agent over 5 years, and has found that 8% of the patient died in the treatment group and 12% died in the control group. So the difference between the control and the treatment group is 4%. Number needed to treat it is 100 by 4, that is 25. So to prevent one death, you have to treat 25 patients minimum. So this is the number needed to treat. The mathematical expression of number needed to treat is given here. Basically, we are looking at two things. One is the outcome, which could be the death, which could be the disease-free survival. And on the other hand, we have treatment.
चारू
And
विंडो अभी खुली है पहले की खुली है नहीं अभी इवनिंग मॉर्निंग से
it's a request. It's a
एसी
request.
चल रही
Please
थी
switch off your microphone before you talk to your family members. Please switch off your microphones. So we are looking at the treatment and the outcomes. And if the treatment is given and the outcome is present, then it is the box is called A. If the treatment is given, outcome is absent, it's called B. Treatment is given and treatment is not given, but yet outcome is present is C and D. And based on that, we find out the experimental event ratio, control event ratio, then absolute risk reduction, which is nothing but the control ratio minus the experimental event ratio. And 1 upon absolute risk reduction is the number of needed to treat. So in most of the clinical trials, you are going to come across the absolute risk reduction, and you are also going to come across the number needed to treat. Intention to treat, most of you know that it basically means that all patients who are enrolled in this study and randomized are included in the analysis. And the dictum here is once randomized, always analyzed. Opposite is as treated analysis. As treated analysis, those who have completed the treatment are only analyzed. Protocol analysis can be intention to treat or as treated. It has to protocol analysis is when the type of analysis is specified in the protocol. It could be as treated or intention to treat. Now, what happens in intention to treat? It takes all patients into account. Any deviation that may happen after randomization, if there is a protocol violation, patients who has not completed allocated treatment because of any reason, like he's lost to follow-up, or he withdraws from the study, he is non-compliant to the treatment given to him, or he refuses to get treated. All the conditions, he will be still be included. This type of analysis is very important when we are talking about minimal invasive surgery, because most of the studies that you are going to read on minimal invasive uh, do not include the cases which are converted into analysis. It has to be remembered that those who are converted are also part of the initial sample, and they should also be included in the analysis, because conversion rate is one of the parameters that tells you that how successful the laparoscopic treatment had. Then there is something called as an interim analysis. This interim analysis is again something which you have to plan when you are planning a clinical research. This interim analysis is an analysis of data that is conducted before the data completion has completed. So that means somewhere half the mark. You may say that the moment I have 30 mortalities in my group, I'm going to do an interim analysis. Or 24 months after recruitment, I'm going to do an interim analysis. The advantage of interim analysis is that if you are going to do an interim analysis, you're going to find out whether the study results are beneficial or harmful. If they are harmful, you can stop your clinical trial in between rather than completing the clinical trial. So this is the advantage of interim analysis. This you are going to find in most of the new clinical trials which are being designed. An interim analysis is planned. Suppose I plan a final analysis, say at 10 years of follow-up, but I can always do a interim analysis when 50% of my recruitment is over, and then see that what is happening. Are there any side effects which are coming to the patients because of my treatment, and then stop the treatment. Now, in a clinical trial, there are primarily 6 stakeholders. There are sponsors who are sponsoring this study. Mostly, these are mostly the people who have the pharma companies who are putting up the money for your clinical trials, investigators, people like me and you who are conducting these studies. Then we have monitors. These monitors are in between the sponsors and the investigators. They are independent, and their job is to do the data monitoring and see whether the data collection is proper or not. Then there are regulators, which are drug authorities. In India, Drug Controller Journal of India is the regulator. or Government of India is the regulator. Then there are patients, and then there are administrators. Like in Banaras Hindu University, if I'm conducting a clinical trial, the administrators or the administration of Banaras Hindu University is one of the stakeholders. So there are six stakeholders mainly. There could be more. So the purpose of research is to find evidence, and all research is based on hypothesis. So why do we want to collect this evidence? We want to collect this evidence because this evidence is going to change the healthcare practice. You will see the fourth line. If there is already evidence and strong evidence, no further research is required. This was a dictum which was being told till some times ago. It has been stricken off because this is something where the research is now taking place. And had the established hypothesis or theory is challenged, we are not going to come across the new data and the new evidence. It is quite possible that the evidence which has been gathered not suitable as to present time because the science changes, evidence changes, the units of measurements changes, everything changes. So with time, as the measurements changes, the collect the data, the instruments that we use, the technology that we use changes, and these changes can probably change the hypothesis. The best example that I use for telling you that how the concepts, established concepts of their time change is gravity. The first concept of gravity was given by Aristotle, who says that gravity is proportional to mass. Challenged by Galileo in his Leaning Tower of Pisa experiment, where he threw two balls of similar size but different masses and proved that the time of descent is independent of mass. It is the size of the ball which determined the descent. Newton gave universal law of gravity that you all know, but it was Cavendish who found the gravitational constant. Until 1798, when the gravitational context of the G was discovered, the Newton's theory was actually was hypothesis. It became a universal law and mathematically proven only when Cavendish gave the G. In 1915, the Einstein gave the theory of general relativity. This is still a hypothesis because what is proven by Cavendish experiments in 1798 is true till now. But there are two hypotheses which has been floated after that. One is Einstein theory of general relativity, where he says it's not an ordinary force, but it is a property of space-time geometry. And Einstein basically talked about from the two-dimensional gravity, which was basically dependent upon the mass and the distance, four dimension where he included the space and time. Now this has been changed by Friedman and Ferrara, who was now floated a theory of supergravity. In supergravity, what he says that gravity is just like one of the fundamental forces, like electromagnetic force, weak nuclear forces, and strong nuclear forces. And when these three forces are included, then it becomes a seven-dimensional theory. And today, the current theory is a 11-dimension theory. And then more and more dimension and more new hypotheses are being generated and added. The last two theories, the Einstein's theory of relativity and the theory of supergravity, Einstein's theory of gravity and supergravity has not been established. But one day it's quite possible that these theories also, these hypotheses also may become mathematically proven. So let's switch to clinical trials. There are four phases of clinical trial, you all know. Phase one is what is known as first in man studies. It is basically a pharmacological trial where you look at the pharmacokinetic and pharmacodynamic of a drug, that how the drug behaves once it is given inside. It is usually done in a normal healthy individual, but the drugs which are toxic or cytotoxic like anti-cancers, these are done in terminally ill patients who are going to die. Phase two are exploratory trials where you basically try to find out the toxicity. So toxicity is to be looked first before you look at the efficacy of the drug, which is done in the phase three or confirmatory trials. Phase four are the post-marketing trials where you try to, after it has been approved, you try to see that new toxicity or new things are not coming up. Little bit about ethics. I'm not going to go in details of ethics, because ethics itself require a full lecture to be taken. So I'll just say that it is a universal principle. There are four principles or four pillars of ethics: autonomy, beneficence, non-maleficence, and justice. So basically, it is talking about that you do no harm to your patients and always take consent from the patient. The consent should be an informed consent where patient is informed that what is the side effects of the procedure or the effect of the procedures, what he gains, what he loses, and the participation has to be voluntarily. So after the information is given to the patient, he decides whether he wants to participate or he doesn't want to participate. There are a number of places where you are going to find, and there are few guidelines which are available. One is a good clinical practice guideline. This is the Indian guideline is available at CDSCO website. Then there is International Conference of Harmonization (ICH) guidelines, which are specific to the pharmaceutical products and how they should be developed. Then there are ICMR guidelines on ethics, which are available on ICMR website, and Declaration of Helsinki, around which all these ethical principle guidelines and good clinical practice guideline or ICH guidelines are built. The Declaration of Helsinki has been modified a number of times, and you can download all these. And I'll suggest that if you are interested in clinical research, these are the four frameworks which you should must read. Now I'm going to introduce three words: law, regulation, and guideline. These three you are going to come across when you are working. Law is a rule of conduct enforced by controlling authority. Like the law which is followed for clinical trial in India is Drug and Cosmetic Act of 1940 and rules of 1945. This is a law. Any violation of the law is a penal offense, and you can be prosecuted for violating the law. Regulation basically is a interpretation of how a law is implemented. So if I say the Drug and Cosmetic Act is the law, then the regulation that deals with the clinical trial is called Schedule Y. There are various schedules: Schedule H, Schedule M. So whenever you are going to pick up a tablet pack next time, turn it around and see which schedule and under which schedule that particular medicine or a combination is governed. Trials are governed under Schedule Y of the Act. And guidelines are basically interpretation of regulations which has no legal binding. So if you do not follow the guideline, nobody can prosecute you for not following the guideline. So guideline is something which tells you that if you are in a particular situation, this is how what you should do. Like the ethical guidelines are basically tells you that this is how you should go about taking a consent. But then you have to actually follow what is given in Schedule Y. If Schedule Y says this is how you take the consent, that is what you have to follow. Guideline may say something which usually says what is with the act, but sometimes it may have something different also, because the guideline can be older, the act can be newer. Like Schedule Y has a change in September 2019, and ethical guidelines have not changed accordingly. So there could be a discrepancy. Whatever is new, if the act is new, you follow the act. This act is available on NACO as well as the CDSCO website. If you have to do clinical trials, please read the Schedule Y, and especially the Rule 122, which governs that how clinical trial should be conducted. So I'm not going to go into the details, but I'll just tell you the amendments. The major amendment came in 2005, and this amendment basically included permission for trial. Before this, it was not required to take DCGI permission for a trial. 2005, they included that every trial has to be approved by DCGI. It also fixed that what are the responsibility of sponsor, responsibility of investigator, and responsibility of the ethics committee. It also introduced informed consent. Before, prior to 2005, there was no concept of informed consent in Schedule Y. This was 2005, it has been brought in, and 2013, this has been converted to video of the informed consent. that the whole process of informed consent has to be videographed. Then studies in a special population like children, pregnant women, and nursing mothers are also included in 2005 amendment. This is a very busy slide, uh, and I don't want you to remember everything. But what are the clinical trials? What are the permissions to conduct do the clinical trials? What are the compensations in cases of trial-related injury or death? Who has the right to inspect? Who gives the permission? The how the ethical committees are registered. These are some of the things which are incorporated into Schedule Y. Now, this one is a very important slide for everyone who is doing a clinical trial or who is thinking of doing a clinical trial. What is considered as injury or death due to clinical trial related injury? If there is an adverse effect of an investigational product and patient dies or develops an injury, it is considered as clinical trial related injury or death, and patient has to be compensated. Suppose you have a drug and patient develops a drug sensitivity reaction. Drug sensitivity reaction leads to or extravasation of drug leads to necrosis or uh, cellulitis, which produces an injury and disables the person for a certain period of time. Then the CDESCO is going to compensate that patient financially, and you are liable to pay that money to the patient. If there is a violation of the approved protocol, scientific misconduct, negligence by either the sponsor or the investigator, patient has to be compensated. Failure of investigational product to provide intended therapeutic relief. So if you are claiming that drug A cures disease B, and if it does not cure it and patient dies, then you have to compensate the patient. Use of placebo in placebo controlled trial. If you are using a placebo in a placebo controlled trial, in certain situations it might be all right. Like suppose you are working on mild pain and you have a mild painkiller, comparing drug A with placebo, you may be all right because if the pain doesn't get control, you can always cross over the patient or give him another drug. But if you are doing a placebo controlled trial, say for example in cancer, you are primarily denying him the treatment of cancer. So in such a situation, if person dies because patient doesn't get treatment of cancer or treatment of hypertension or treatment of diabetes, you are liable to pay the loss and damages to the patient. There are adverse effects due to contamination, concomitant medication excluding standard care necessitated as a part of approved protocol. So if you give a drug and the drug, additional drug has a reaction, you are liable to pay. You didn't do a pregnancy test, included a woman in the trial, she becomes pregnant and the uh, child in utero dies. You have to pay compensation. There are a number of ways this compensation is given, and the Schedule Y, the new guidelines which are come in 2019 also defines that how this compensation is calculated. I have not included it here. So probably when we are talking of Schedule Y and clinical trial regulations, I'll probably talk about how the compensations are calculated. Another important thing for all of us to remember that the ethics committee, which is reviewing the protocol and providing approval for the clinical work, clinical trials, should be registered with DCGI. So if there are centers, because now there are more centers working and all thesis work is going on, all thesis work is considered as clinical work, especially in the clinical sites like surgery, medicine, clinical branches. And you get an ethical committee approval, you have to ensure that the ethical committee is of your institution is registered with DCGI. If it does not, they cannot accord approval to the study. New drugs CTR rules 2019 .pdf is the PDF available. It's a gadget notification, and you will get it from the CDESCO website. So what are the prerequisites for running a clinical trial? You have to have a permission from Drug Controller General of India. You have to have approval from your ethics committee where the study is planned. Then the clinical trial has to be registered on ICMR mandated maintained website. This is ctri.in, and you should have insurance. Now, the insurance has become very important component because uh, uh, the compensation part has been added. So you may be required to pay compensation, and hence the insurance part has become very important. Monitoring. Uh, 2013, there is a very famous case which is going on, which was filed in Supreme Court, was Swasthya Adhikar Manch, and this case made a lot of uproar. If you go back to 2013 newspapers, you'll find a lot of unethical clinical trials and deaths from the clinical trials being discussed. And after that, the Supreme Court has directed that there should be a monitoring panel. There are two monitoring panels. So uh, the first monitoring is actually done in your own institute by your own ethical committee, which I'll come to later. But there are two committees which are monitoring every clinical trial that is conducted in this country. One is the technical committee, which is under this chairmanship of the DGHS, that is Director General of Health Services, and then Apex committee, which is chaired by the Secretary Health and Family Welfare. So these committees meet regularly and they evaluate and monitor the progress of every clinical trial. The ethical committee, one has to submit a report every 6 months of the progress of the work which has been done. And apart from that, there could be a local monitoring committee which can monitor which can go to the site, go to the place and monitor. And then there could be a monitoring by the trial sponsors, which I already talked about in stakeholders. So there are three local monitorings: monitoring by the local monitoring committee, monitoring by the ethical committee, monitoring by the trial sponsors. Then there are two monitoring bodies, one under chairmanship of DGHS and one under chairmanships of Secretary Health. So all these reports are, when the report is submitted, suppose there is a severe adverse event or an adverse event. Every time an adverse event happens, a report is given to the ethical committee, who deliberates it in their meeting, files a report on it, and then the ethical committee sends it to the DCGI for placing before the uh, DGHS committee. And from there it goes to the health secretary committee, and if they decide that the severe adverse event or a death has occurred because of this, the compensation will be awarded. So what are the problems in conducting clinical trials? One is complexities. There are a lot of regulations and every year it is becoming more and more complex. The cost required to conduct clinical trials is huge, because the insurance component has now come in and the compensation component has to be taken into account. The cost of clinical trials have increased. And patient access is another one. The moment you tell somebody that it's a new drug, may benefit, may not benefit, the chances are that patient may or may not agree to participate in clinical trials. In United States, it is permissible to advertise participation in clinical trials. So you are going to find advertisements asking people to ask their doctor for eligibility into clinical trials. But in India, it is not permitted so far. Then the staff role and responsibilities has been very well defined now, and it creates a lot of burden on people because it puts extra burden, extra work, extra paperwork into everybody's basket. Governance and oversight and new drugs are other problems. So a little bit, just introducing some other terms which are associated with clinical trials and the publication of clinical trials. One is misconduct. Now, misconduct basically is intentional wrongdoing. People will always ask what is the difference between a mistake and a misconduct. So the difference between mistake and misconduct is intention. Anything which is done intentionally is misconduct, and if it is done unintentionally, it is a mistake. This is one thing that you have to know, that misconducts. In research misconduct, the definition says fabrication, falsification, plagiarism, in proposing, performing, or reviewing research, or in reporting research result, is all called research misconduct. Penalty for research misconduct: in Banaras Hindu University, if you are a student, you are terminated immediately. And if you are a faculty member, then also you are asked to uh, leave the institution if the research misconduct is proven. Very severe penalties are imposed by regulators, the administrators these days. Now, there are various ways that why data may be incorrect. If you have a wrong design or an inappropriate assay, your data could be wrong. If your hypothesis is wrong, which is often based on the other people's work, and if their work was wrong, then your work is also liable to be wrong. Bad instruments is one of the commonest things. It is just like your bathroom weighing scale. If your weighing scale is +5, then everything measured on that will be +5. So if your instrument is not accurate, if it is not measuring accurately, all measurements made by that instrument will be wrong. Inappropriate analysis of data. This is something which we have been discussing, that how data should be analyzed, what tests should be used. If interpretation of data is wrong, then also the research is wrong. And then bad ethics and scientific misconducts is also participating to in incorrect scientific data. A new thing that has come up for quite some time is now conflict of interest. Conflict of interest is a set of conditions in which professional judgment concerning a primary interest, which is the patient's welfare, tend to be unduly influenced by secondary interest, which is a financial gain. This is more commonly seen in the West, where a person who develops a new drugs also get kind of shape by shares in the company or the shareholder in the company. So if a drug is approved, he also benefits from the rise in the cost of the share and its participation in the company. So financial gains. Second thing is that some of the investigators are funded by the company. So they may be doing a clinical trial which is funded by the same company who is manufacturing that clinical trial. So this conflict of interest has to be declared that the principal investigator was funded by the pharmaceutical company. New thing that has come in science is whistleblowing. Whistleblowing is that another member of the organization comes out and tells people of the illegal doing or immoral practices which are happening in the institution or by their colleagues. So it is called whistleblowing. It is same as whistleblowing in politics. Coming to the some of the last slides, that why do people cheat? One of the important thing is that there is an intense competition. Either you publish or you perish. So if you are not able to do the research, if you are not able to generate the fundings, if you are not able to generate enough amount of fundings, you may lose your job. So publish or perish is more pertinent to India, where for promotions you are required to publish 2 original articles. So if you are not going to do research, you will not be able to come out with 2 original articles. So you have to do some kind of research and produce some kind of 2 papers. Loss of funding is more appropriate for Western world, where the especially US, where the salaries are linked to the R0 or R1 grants that a person receives, or his laboratory funding and his supportive fundings. Everything is dependent on the amount of funding that he receives from the funding agencies. It's not very pertinent to India, where you get your salary irrespective of the research that you do. Then there could be greed. People may do it for money, people may do it for fame, people may do it for awards. Some people are so sure, they are so arrogant that no, they say that if I do this research, this is the only outcome that can come. So arrogance can be there. And most important is the cultural differences, where the values and definition makes values in the society makes a huge impact. So we know there are certain societies now. If you look at the publication ethics, or I also edit a journal, so if I look at the journal submissions and the review reports, the most scientific misconducts are being conducted in China, which is at number 1 at present, followed by Iran at number 2. India is not far behind. The impact of this scientific misconducts, what happens? Who are the people at risk? Most importantly, me and you, because we get cheated. Somebody has falsified the trial records and a drug which was not working has been proven as working, it is taken by me, you, our family members, and we all end up suffering. There are a number of drugs which has come to the market and has been withdrawn. The COX-2 inhibitors is the recent story which increased the incidence of stroke in patients who were using COX-2 inhibitors. Then the honest scientists who are actually doing the work, their reputation is also tarnished. When I said that two countries from where the maximum scientific misconducts are being reported are China and Iran. Now, everybody in China is not dishonest. Everybody in Iran is not dishonest. But what happens is that even when there is a honest research coming from those countries, that also goes through a microscope to see whether this is properly conducted or not conducted, ethics approvals have taken or not taken, consents are there or not there. So even the honest scientists who are working on those countries are also sufferers. Third, where does the money comes from for this research? It is the taxpayers' money. It is out of the money which is allocated from the GDP on the healthcare. So it is our money or the public money which is wasted. So these are the three people who are basically sufferers. Now, the best way of avoiding all these trouble and streamlining thing is education. But unfortunately, clinical trials and clinical research is not in the curriculum, and nobody teaches how to conduct a clinical trials or how to do a research. So, in absence of a defined curriculum, BHU has introduced research design, ethics, and clinical trials in PhD curriculum, but it has not yet included it in the MS, MD, and MCHDM curriculum. Second thing is mentoring of the students. Basically, the people who are doing research, the students learn how to do a research. If a person is doing a honest research, his students learn to do honest research. If a person is doing a dishonest research, his student learns to do dishonest research. And now there are some web-based classes and courses have started, and these are also available. So everybody does it that way is not an excuse. So you have to be different from what other people says. So I'll end the talk with conclusion that clinical research should be conducted strictly as per the guideline, rules, acts, and regulations that everyone has to read and know. Violation is punishable offense, and one can have even a jail sentence for violating the clinical trial rules, laws, and Schedule Y. Application of proper research design, analysis, and interpretation is essential because if any of the three things is wrong, the research outcome will be wrong. Always keep your local ethics committee in the loop. If there is any adverse event or SAE, inform your ethics committee within 24 hours that this has happened and keep informing them, updating him till the patient dies or goes discharges and goes home. This is very common in oncology clinical trials because most of the people who are given the uh, anti-cancer drugs develop neutropenia. So the moment there is a neutropenia in a clinical trial patient, within 24 hours, inform your ethics committee that there is a neutropenia and start taking action to correct that neutropenia. The patient, the moment the neutropenia is corrected, patient is fine. Again, inform your ethics committee that this was there, this has been done, and now the patient is discharged and gone home. Take consent, keep patient informed, be ethical, and avoid trouble. Thank you.
Sir, if
Yeah,
I may ask a question,
yeah.
because nobody else is asking, I thought I should
No, it's
ask
okay.
the question.
Yeah,
It was in my mind for a long time.
yeah.
Sir, being a teacher in a medical college, we surgeons are pretty busy with our surgery, with our clinic, and everything. I'm not talking about the MCH students because they are always in the department. We have general surgery PGs, and we are their guide and co-guide. They are rotating through different units. So the problems we have is we are actually but sometimes every PG is different and how much the research matters to them because again publish or perish, they have to do their thesis and get it out for the post graduation. So how do you keep think finally you are responsible for the ethical and for the final result and publication, whether the data is appropriate or not. So what are your methods you would suggest to keep them in check and be very careful about it?
See, the commonest thing that the postgraduate students do is manipulate the number of cases. They take less and make it more. They may multiply it by 2, multiply it by 3, multiply it by 4. If you are quite careful when you are going through the data, you will be able to find out that
Yes.
how what they have actually done. You have to go
Sir, we
carefully
have
from the
the
data, cross check if possible. What we normally do is, I think you also must be having is a pro forma.
yes,
So what
yes, yes,
we normally do is, now you have to remember, Anshika, that these pro formas which you get filled up have to be kept
yes,
with you for at least 10 years.
right.
You have to store them for 10 years. So normally what we do is, we ask our students to fill up the pro forma. We take up the mobile number of the patients in that pro
Right.
forma. We have included that as a field. And anytime you feel that a particular pro forma is not filled up properly, or if the data could be wrong, you can just
Hmm.
pick up your
Hmm.
mobile phone, call that person, and find out whether the person and the mobile number is correct or not. So if you
Yes.
have a check
So
mechanism,
we have the UHID. We have a UHID, hospital
yeah,
identification
yeah,
numerical digit.
yeah.
We personally check each and every UHID whether it exists or not and all
Mm,
that.
mm.
But my worry also extends to the details in the data. Uh, the outcome things you measure in each patient. So it's always a ongoing struggle for everybody, I guess, to make sure the data is
If you start, I don't know who did your analysis. Like if you do your analysis yourself, you will be start identifying that when the person is cheating in a minute. It's not very difficult to actually go through the data and find out. So it comes by experience. You know a false data when the moment you look at it. You know that this is not possible. Some or the other parameters do not match. What we do in a social science, and sometimes we do in clinical research
Hmm.
also, is we keep
Yes.
cross-checking questions. So
Right.
if you have uh, you frame two questions which are actually looking at the same thing from two different angles. Say
Right.
say
Yeah.
you ask the three-dimensional size of the tumor, and then you ask T stage.
Correct. Correct.
Okay, so if you try to keep these two questions for key parameters, you probably are going to end up identifying whether the boy is doing the cheating or not doing the cheating. So this is the only way you can. Otherwise, it's very difficult. Boss, the ideal way of choosing is that you go through the back issues of that journal, see what kind of articles they are publishing, and compare it with the type of articles you want to submit, and see if they are considering that kind of articles or not, or go for your specialty journals.
Like Copernicus, something like Scope, ISI. What are these things?
Sir, basically, what the government of India wants is a indexing in one of the indexing services. The commonest is the PubMed or the Medline. Apart from that, the second which is acceptable is Scopus, and one is which is accepted in India is Index Copernicus. These three indexes means these are the databases where your journal name is written, or your journal. Now impact factor on the other hand, or uh, citation index which scholar gives you, is something different. That basically looks at that how many times your article is cited. So indexing is different and impact is different. This RG index is not recognized by government of India.
Observe our negative results also. No? That should be.
Sir, negative results any journal will publish. That is not a criteria. It is basically the design and conduct of the study and the level of analysis that you do, which is important. Negative results most journals will publish.
So these sir, if you look at the journals, some are online journals only, and some are online as well as print. So as far as possible, we should not, because we are doing investing so much of our time and energy in the clinical trial or study which we are doing. So preferable is to avoid the only online journals which don't have a print copy.
No, Anshika,
Sir, what do you say?
that is wrong actually. Actually, what is recommended by the NCI in US is to go for open access, because the government of United States says that any study which is funded by the people from the funds of the people of United States should be made available to each and every citizen of the world. So actually, they are promoting open
Sir,
access.
open access. Sir, open access and versus non-open, I would
Yeah.
say is when you want to access the journal, you don't have to pay an amount to access that particular
Open
article.
access is
But I
always
was
online.
referring.
Open access is always
Yeah.
online. Open access cannot be a
So
print
what I
journal.
was suggesting is.
Yeah, most of the people have most print and online, but if you go to Springer Online or
Right, sir.
Nature Online,
Right,
so
right.
even Nature Group has online journals only, because nobody wants to go, nobody buys a print copies nowadays.
So then, why the government of India and MCI is hell bent on
Government of India
the
has
print
its
journal?
own.
What is that, sir?
Government of India has its own. Sometimes it relaxes too much, and sometimes it becomes this thing. Most of the journals will have a print copy. They don't print it, but then the print copy is always there. The PDF you are going to get.
Yes, sir. Yes,
So,
yes.
so they don't print. Most of the journals are not printed. See, even
And I
the
agree, sir. Open access is the best and the most fruitful, because then people can actually read your article
yes,
in whole,
yes,
not just the
yes.
abstract. Yes, sir.
And look at IJSO, our surgical oncology journal. Journal of surgery back issues in my home that I don't know what to do with them. Even the libraries are not willing to take them now.